Good evening to the Artist Now lecture series. So thanks for coming. And in a second, I will introduce Jennifer, who will introduce our guest. But I want to highlight next week and invite, obviously, everyone back to the lecture next week, which is Arthur Hash. And the statement from the Artist Now brochure reads, Hash discusses how today's craftsmen have the freedom to choose from any method of making or material that is available to them, giving them the ability to blur the boundaries of craft and redefine their fields. Sounds like an object guess, right? So please, we have a different field of study, a different artist next week. Spread the word. Obviously, there's many students in this course that'll be here, but we should really promote this series to our students better. Could I argue that? This is one of, hands down, our best lineup. I say every week, every time you have a chance to see a visiting artist, it's always vital, it's always interesting. You always learn how artists live, what type of work they do, what type of opportunities are out there in far corners of the world. Tonight's guest is coming from Helsinki tonight, and his work looks absolutely fascinating and it's groundbreaking. We should have this lecture close to full every week. And that falls on the teachers and the students. Yes, the students, the art students. So please tell your fellow students and teachers, tell your students, require is a hard word, right? But urge people to come. Because why have a visiting artist lecture series if we can't fill the audience, in my opinion? So this is hands down one of the better lecture series that happens in the state, if not the Midwest. So the lineup's fantastic, but we shouldn't have empty seats. We should have a full house. So please, many of you use social networking nonstop. You can promote this to your friends. This is free and open to the whole public. So that's my spiel. I won't give it again, but I think we should be promoting this and showcasing these incredible artists in this series that's absolutely vital to the intellectual growth, the cultural growth, and artistic growth of the city. Right? Yes. Yes. So come on, let's fill it up. So at any rate, I am going to introduce Jennifer Johan, who is going to introduce tonight's guest. Jennifer.
We are incredibly lucky to have Oron Katz here with us today on the way from Helsinki, where he is currently setting up a biological art lab called Biophilia, Base for Biological Art and Design at the School of Art, Design and Architecture, Aalto University, Helsinki, where he is a visiting professor. He is also the founder and director of Symbiotica, the Center of Excellence in Biological Arts, which is housed within the School of Anatomy, Physiology, and Human Biology at the University of Western Australia, and he's also a visiting professor of design interaction at the Royal College of Arts, London. Oron has also been a research fellow at the Harvard Medical School and a visiting scholar at the Department of Art and Art History at Stanford University. He's an artist, researcher, and curator whose pioneering work with the Tissue Culture and Art Project, which he established in 1996, is considered a leading biological art project. In 2000, he founded Symbiotica, an artistic research center. Under Oron's leadership, Symbiotica has gone on to win the Prix Ars Electronica Golden Nika in hybrid art in 2007, and became a center for excellence in 2008. In 2009, Oron was recognized by Thames and Hudson's 60 Innovators Shaping Our Creative Future in the category Beyond Design, and by Icon magazine as one of the top 20 designers making the future and transforming the way we work. Katz's interest is life, more specifically the shifting relations and perceptions of life in light of new knowledge and its applications. Often working in collaboration with other artists, mainly Ionit Zur, and scientists, Katz has developed a body of work that speaks volumes about the need for new cultural articulations of evolving concepts of life. Katz's ideas and projects reach beyond the confines of art. His work is often cited as inspiration to diverse areas such as new materials, textiles, design, architecture, ethics, fiction, and food. And he has no doubt a very provoking presentation for us today. So please join me in welcoming Oron Katz.
Thank you. Thanks so much, Jennifer, and it's really great to be here. And thanks so much to everyone who's responsible for bringing me all the way from Helsinki, or actually Australia via Helsinki, but that's a different story. What I thought about doing with you today is telling you some stories. It's not going to be a traditional artist talk. I think it will be quite different than what you're usually accustomed to. But I think there's some really important things that we need to go through. In the mid-90s, I was actually studying design, and that's why I have those design credentials that I seem to receive from some strange reason. And I came across a paper by a design theoretician called Ezio Mazzini, where he was talking about the need for us to think about a new made environment, one in which we care for the products that are becoming part of our world. He said he wants to think about objects not as instruments for use, but as entities that are effectively linked and that need care. Think about objects as plants in a garden. Think of objects that are beautiful and useful as trees in our own garden, objects that endure and have lives of their own, objects that perform services and require care. So he was thinking about a criteria of quality that leads to a system of objects that have the variety, complexity, life and blend of beauty and utility of a garden, but at the very same time are a product of the real world, a world extensively and intensively artificial. He was talking about those smart objects that we see around us now, but I took it literally. And I decided to see if we can actually grow objects like plants in our garden, trying to change our culture from a culture of manufacturing to a culture of growing, trying to create a culture where we would start to care for our made environment because it's going to be alive. As I was thinking about it more and more, I realized that also the flip side might be correct as well. If we are starting to make the living environment around us our made environment, we might care less for the living. And I was really interested in this paradox and the tension between trying to think about growing stuff around us, as well as starting to treat the growing stuff as something which is man-made. And I think, in retrospect, that was mid-90s, I realized that we are moving into a world where biology is increasingly becoming a form of engineering as opposed to a knowledge production. So, and it goes back to quite an historical perspective. So if you think about what happened even as early as the 1750s, where you started to see that our relationship to the living became more and more industrialized. So this is what actually was called the artificial mother. This environment in which we can start to grow chickens and fowl in a way which is very similar to the idea of the factory. And the idea of the factory is extremely important because when Ford was thinking about the assembly line, he was actually influenced by going and seeing those disassembly lines in Cincinnati and other places, which was really the birth of this idea of the division of labor, the idea of the fact that when you start to take the living apart and treat it in a manufacturing, in an industrial way, this idea of manufacturing farming or manufacturing life is becoming very apparent. So this idea of the application of engineering logic to life, as I mentioned, wasn't, it's not a recent thing, but obviously it's increasing now to such an extent that I think that we all need to pay more attention to. But when you think about a statement out of A.G. Wells in 1895, where he was talking about the fact that life is a raw material, something to be shaped and altered, something plastic to be shaped and altered, you might find it disturbing. Well, how many of you find it disturbing? That's good. All right, good. Because obviously, and that's, I use this quote for more than 10 years now, and the amount of hands that are being raised is becoming increasingly rare. People are accepting the fact that life is becoming a raw material, something plastic for us to shape and alter. And again, when H. Wells wrote it, he was influenced mainly by this guy over here, Jacques Lau, who was really dreaming about this idea. And again, it's late 19th century, beginning of the 20th century. He wanted to see an engineering biology, a biology that is prescriptive rather than just descriptive, and engineering, or a biology which really employs engineering logic in order to drive the way we treat the living. And his work, which was dealing with the artificial creation of life, seems to be kind of coming back recently through things like synthetic biology. So I would argue that, first of all, the way we think about life can be easily measured by how we see industrial farming or factory farming. And we're going to see that some of you might come to the SLSA meeting, there'll be quite a lot of discussion around it. But I really like this quote from Jonathan Safran Foer, where he was talking about the fact that for industrial farming, for factory farming, nature is an obstacle that needs to be overcome. Right? So we're starting to think about life, we're starting to think about nature, not as something that we want to share life with, but something that we need to overcome, some kind of an obstacle that will enable us to have this degree of control over life that is required by employing this type of engineering logic. And you can see that the language of the engineering logic is apparent. That's again the early 20th century. Cypher's incubator, the first industrial scale chicken incubator, so what you've seen before, the 1750 was a bit more backyard hobby farm. This is really the standard of the world. And when you think about standardization in the context of engineering, it's very, very important. And obviously, that's the reason to have it, the profit. So, alongside the time where those industrialized ideas of dealing with life came about, also this guy over here, Alexis Carrel, was starting to think about this idea of how we can maintain life in ways which were impossible before. And he came up with a technology, or an area of research which is called tissue culture. The idea that you can actually fragment the body of the complex organism and maintain parts of the body alive outside in artificial conditions. And in a sense, by that, creating a new type of body in which to grow those fragments, the technological body. Now, when you start to think about those things, they're really weird. The idea that you can actually keep parts of bodies alive within those artificial environments is something that akin to maybe a freak show. And that was really the case. So here, early 20th century, the first baby incubators in the United States, this is one of the first premature baby wards in the United States in Buffalo. This is the one in New York. Spot those crowd control rails over there. Because it was in Coney Island. You can see here, the infant incubators with living infants. People would pay money to see those freak bodies in incubators. The combination of living bodies and machines was something that could only resolve itself within the notion of the freak show, because there was no cultural articulation of what's going on. And I would argue that we're living in a very similar situation at the moment. And when the cultural articulation is coming to bear, there's some really dangerous stuff that might be generated from that. So you can see another piece of work by Alexis Carrel, but also this guy over here, and remember him, Charles Lindbergh, your great American aviator, one of your great heroes. Because those people were designing this organ perfusion pump, this artificial heart, to a large extent very similar to those chicken incubators. This is an instrument that was designed for display, not just for science. And you can see it was exhibited as a piece within the World Fair. This chamber of life, if you like, the chamber, and actually it was referred to as a chamber of artificial life, was held as a starting point, and to some extent it is, to the whole science of transplant. And the interesting thing was that when, and here you can see actually Lindbergh and Carrel working on the organ perfusion pump at the Rockefeller Institute in New York. And this is the cover from Time magazine. This organ perfusion pump, this chamber of artificial life, came about from Charles Lindbergh, who really was a mechanic, thinking about the fact that when something in his plane breaks down, he takes it out, fixes it, put it back into the plane is flying again, and he goes to Carrel and says, how come we can't do it with the human body? The human body is a machine. Let's try and fix it, let's try and do it in such a way. And that's what brought them about together. Now, they also shared some ideological similarities, and I will talk about it a bit later, but just note that in the same time that Carrel and Lindbergh were working on the organ perfusion pump, Carrel published a book called Men the Unknown, which we'll discuss shortly after. Now jumping about 60 years forward, and this idea of the body as a machine was obviously very much apparent in the ideas of how we thought about repairing the body. So this is from 1989, from Life magazine. You can see the idea there was that if something breaks down in the body, we would just develop the technology, we would develop a mechanical apparatus to try and solve it. Comes 1999, 10 years later, everything was twisted. Because the discovery or the conceptual shifts about bodies that came about through this new area of research, which is now being referred to as regenerative biology or regenerative medicine. The idea of trying to harvest the latent regenerative powers of the body in order to replace those body parts. So this is kind of what I was referred to as the Wizard of Oz Tin Man approach, and that's kind of this amphibian regenerative salamander approach. So the whole idea, the whole shift towards regenerative medicine came about through this new technology that was developing in the mid-90s, and those of you who noticed the mouse with the human ear growing from its back, that was one of the poster kids of this new technology. And this field of tissue engineering was based on the assumption that one would be able to create a three-dimensional structure out of a special degradable material, seed it with the appropriate cells of the organ you're trying to replace, and then grow it in culture outside of the body for a while, and then implant it into the body, and you have a new organ. That was the proposition. In 1995, when the first paper of tissue engineering was published, in 1993, there was the proposition, and those people who were developing this technology were promising us that in about 10 years' time, then, we'll be able to have a fully functioning heart being grown outside of the body. As I was doing product design at the time, my question was reverse. If we were told that in the 80s that we can replace the heart with a pump, come 90s we are told that we can actually grow a heart, my question was, why not grow pumps? Yeah. So forget about that, forget about the human body. What about creating those structures which are partly alive and partly constructed, that might bring about this new idea, as I referred to before, of growing things as opposed to manufacturing them. So in 1996, I set up the Tissue Culture and Art project. Yonatsuo joined me shortly after, and we approached the scientists with this idea of using living tissue as a medium for artistic expression. We are really interested not so much in trying to find solutions, but we're interested in the idea that we might need to resolve the complex paradox that we were starting to realize in regard to our relationship to life. As I mentioned before, if we are starting to treat life as a raw material, what kind of relationship we're going to have with it? So the realization was that we should really try and explore it as an artistic pursuit as opposed to any other profession, because art is the most capable profession and the only one that still has a license to engage in paradoxes in an open way. Because of this engineering approach that seems to take over quite a lot of other professions, as artists we still have this freedom to engage in open questions, to seek problems, to fail, and to engage in futility. And that's something that we wouldn't be able to do in any other profession. But with this idea, we knocked on the door of a scientist, and she invited us to come and work in a college lab who was running an eye research institute, or he was running a research program within an eye research institute where he was trying to develop artificial corneas. And we were confronted by something very, very strange. So this is really one of the very first things we've seen in the lab as we approached it, and I have to remind you where life comes from. That can be quite bloody. But what we were quite amazed by was that the research institute was using rabbit's eyes in order to try and see the compatibility of the tissue that grows over the eye, the skin tissue that grows over the eye, with the type of new materials that those scientists were working on in order to create this artificial cornea. Now, as you've seen, those are half rabbit's heads. The rabbits have been killed in the morning for food. The heads would be sent to a brain research institute. The brain would be taken out. And then we would get those half rabbit heads about lunchtime. We would then pull the eyes out. You can see there. And then put them inside those vials with antibiotic solution overnight, and only start to culture those cells more than 24 hours after the animal was visibly dead. So the whole question that we were starting to confront, on top of our questioning about the idea of growing things, was the fact that life and death are not as defined in this context as one is culturally adopted to engage with. And we also were very much realizing that the only type of level of engagement that we can have is in the realm of science fiction. And this is actually a quote that one of the chief scientists within this research institute was telling us as he was walking in a corridor holding this vial full of ice. And obviously he could only engage with us on that level as well. So as we were looking down the microscope, after culturing those cells from those eyes that were taken from a rabbit that was dead for more than 24 hours, we would start to see those cells proliferating and growing, and we were quite realizing that we don't really have either a verbal but also a cultural language to engage with those things. And we realized that we are actually in the Stone Age of biology. That in a very similar way that our ancestors started to realize they could chip away stones in order to create tools, which what created, or what was developed later on into what we are now, a technology-based organism, we are now starting to chip away life in a very similar way that we have no clue where it's going to take us. And we had a series of works, which we call the Stone Age of biology, where we actually use prehistoric stone tools, miniaturize them, and then grown different types of cells over them, and created this piece, or this series of works. But because I really want to tell you other stories, I won't dwell on the very early work that Jonathan himself did from 1996 to 2000, and actually not until around 2001, when we started to also develop a series of works that we refer to as pseudo-utilitarian works. And we were titled the whole series of works as the technologically mediated victimless utopia. We're really interested in this whole discourse of the victimless, of the idea of what happens when you remove the victims of one's consumption, what happens when you start to push away the victims to such an extent that they're becoming invisible. Because in a sense, this is how Western technology operates. This is how you Americans wage war as well. Yeah? You don't want to see the victims of your operations. You want to have a button, you want to sit here somewhere in the States and press a button and drop someone in Afghanistan, yeah? Without this direct connection. And in a sense, this is very similar to what's happening with the way we treat life. So, the first of the experiments that we've done was our in vitro meat project. And
at the University of Western Australia in Perth, a strange creation is slowly growing in a lab. Oron works in the biological sciences department. He's making some of the world's first artificial meat.
When we grow those cells, we actually tend to refer to them, we coin the term semi-living, because they're not fully alive, they're not an animal, but they are a living part of an animal sustained in an artificial environment.
This artificial meat was grown from the muscle cells of a lamb, but they could have been cells from any animal. The cells were cultured in a nutrient solution made up of blood plasma derived from cattle. The muscle cell cultures are grown in a microgravity bioreactor, which constantly rotates the meat so it grows in 3 dimensions. Without this motion, the meat would be flat, only a few cells thick. But there's another problem to overcome if the lab-grown meat is ever to mimic the real thing.
There is research which is being taken place at the moment in trying to find ways in which you can exercise those muscle cells so they would align and have a more of a muscle architecture. And there's 2 ways that can be done. One is by electrically stimulating those cells, or the second way is to try and exercise them physically, trying to build tiny exercise machines, tiny gyms for those cells to grow and become more like muscle.
Oron has brought his unique piece of engineered meat to the UK, to one of the world's great chefs. With 3 Michelin stars, Heston Blumenthal is famous for fusing science and gastronomy. This meat is so cutting edge that in Britain, it's not recognized as food and so can't legally be eaten.
It's got a smell of raw liver to it.
I never really sniffed it, but
I'm not going to eat it, it's about as close as I can get. You're going to turn it into the world's most expensive, tiniest hamburger.
yes, which it is at the moment, and it's as big as one can get at this stage.
Many people may find the notion of lab-grown meat less than appetizing, but not this master chef.
I think as a catalyst for conversation, debate and thought, it's fantastic because how many people are quite happy to eat sliced ham that actually is not ham at all, it's processed meat from animals that have been literally chained to the floor.
Yeah.
That's not a problem, but something that's been grown
in a
lab actually is a problem. To me, that has the potential to be a lot more ethical.
The technology is still in its infancy. Even when scientists figure out how to ramp up production, this lab-grown substance would only work as ground meat. Engineered steaks and chicken breasts are only in the dream stage. And none of it is economically viable to produce, yet. Although artificial meat may be decades away.
Okay, so a couple of things to notice about this video. This video was prepared as part of a 2 hours documentary of the BBC and Discovery Channel just before the Copenhagen Convention, which was supposed to replace the Kyoto Convention about environmental issues. And this whole documentary was about extreme technological solutions to issues that are being caused by human technology in regard to global warming. So, you know, it was an interesting artistic intervention from my perspective. It was also a really nice commission, and it was the third time that I was able to grow a steak. So the first time was when Yonata and myself were research fellows in Harvard Medical School. The problem there was that the steak that we were growing, we couldn't eat because the laboratory that we worked in didn't have a license to grow food. And obviously you've seen it also with the third one, where we couldn't import it to the UK as food, so we couldn't cook it over there. The only time we could actually grow and cook it was in a show in France in 2003, in a big art and biology show called L'Art Biotech, where they had a budget which was big enough for us to go and live for 3 months in the gallery or around the gallery and prepare and build a fully functioning lab that also had a dining room there. And because it was in France, we were also playing on this idea of what constitutes foul food. So we know that French people don't like the idea of engineered food. We know that most other people find the idea of eating frogs quite disgusting. So we decided to combine those two things together and grown our first tissue-engineered frog steaks. And for the 3 months of the show, we would go every day and feed our steak, basically change the nutrient solution. We also, as a gesture, rescued 2 frogs from the edible frog distributor in the town there. So those frogs were destined to be food, and they lived with us in the gallery for the duration of the show. And then in the last day of the show, we had this ultimate nouvelle cuisine dinner. We invited people from the audience to join us for this dinner, and we shared this really tiny steak we were able to grow for the 3 months of our time there. And you noticed also at the very beginning of the video about the meat, you've seen what a large chunk of meat I had to take initially from the lamb in order to grow something as small as Hertz Blumenthal was testing. And it was similar here. We basically took us 3 months to grow about 4 grams of food, of steaks. And then out of the 8 people that joined us for dinner, 4 of them spat out the steaks. Which was great because I could then collect it and show it in the follow-up exhibition called The Remains of Disembodied Cuisine, where we had the bits that were spat out, as well as the 3-screen video installation about the whole process. But also we released the frogs that we rescued in the ponds of the botanical gardens, just for them to be eaten by the ducks later on. But what I want to tell you, first of all, it was an extremely expensive operation. Secondly, in order to grow cells, you need to feed them. Yeah? They can't just grow out of air. And in order to feed them, you need to use animal-derived materials. So in order to grow that tiny 4-gram steak that we were able to grow, we needed to use something like, I suppose you're in the imperial, but anyway, 500 mils, so half a liter of fetal calf serum, which is basically the blood plasma from an unborn cow. So when anyone tells you that tissue culture experiments are a nice, neat way to reduce animal experimentation, or when anyone tries to sell you the idea of in vitro meat as a great solution, you should ask where the food is coming from. How do you feed the cells? And in most cases, and that's a problem, in most cases, there's no solution other than feeding the cells with animal-derived materials in the first place. So it's a really strange protein translation operation that doesn't make sense, besides the sense of hiding the victim ever so further. And that's similar to what we later on did with a series of works around leather as well. So here you can see the Victimus Leather work that became one of our most popular works because it was shown, and this is one of the experiments towards it, but the Victimus Leather was shown initially in 2004, and then in 2008 it was part of this big design show at the Museum of Modern Art in New York. But what happened there was that we were using a new type of cells. So if before, for growing the Victimus leather, we were using a combination of human and mouse cells to grow into this leather-like jacket. For the MoMA show, they didn't really want us to use human cells, and we were trying to figure out what other cells we can use. And we got access to a laboratory where they were working with mouse embryonic stem cells. And those cells were amazing, but it grew so fast that some of them sheared off the actual structure out of this leather-like material, or the polymer, and started to clog the system. So about 5 weeks into the show, they had to switch it off. And this is the headlines. So, New York Times said, MoMA kills art, which is, I think, can take the credit for that. And then murder in MoMA. And basically, this very optimistic design show, which was called Design Elastic Mind, it only had one living piece within it, had to kill that living piece halfway through the show. So it wasn't as optimistic as one would imagine. And from our perspective, it gained this type of responsibility, or the issue of responsibility that one needs to exercise towards living systems that one is involved in manipulating. Yeah? So it generated a really, really interesting discussion, but also from the perspective of us growing the Victimus leather, we were much more interested in where things go wrong than when things go right. So this is another example of something very strange that happened when we had a show in Luxembourg. And it's quite amazing when, as an artist, it works with galleries that I used to have, they used to have dead stuff that never change. And actually they have people that the whole job is to try and make things not changing. And then we're intervening within this system, and we get those crazy phone calls in the middle of the night, usually to do with the time difference. We're back in Australia and say, something is wrong with your stuff. I say, of course, it's alive, it's changing. There's no control. And so we get all of those strange images. When we were showing this work in the Mori Art Museum in Tokyo, we had this tiny infection. It's a fungal infection. And it was really nice. It was a show which was titled Medicine and Art. And after getting the phone call, actually I was still in Japan at the time, I went for the first 10 days and I was trying to administer medicine to my art in a show which was called Medicine and Art. And kind of trying to arrest the development of that fungal infection. But when I left, created this amazing thing, and in a sense I think this is much more powerful than just the human and mouse cells that we're using to grow the cells. And now, just recently, last month, literally, we found out about those people over here. Modern Meadow, the company, that just got money from the guy from PayPal, $350,000 to create 3D bioprinting of meat and leather. And I actually came across them, and they were using this rhetoric of saving the world. And I said, don't bullshit me, yeah? You are engaged in something which is extremely expensive, extremely useless. What you're dealing with is actually a luxury item. It's the kind of the ultimate inaccessiveness of technological discourse and technological approaches. And that kind of led me to think about the fact that we are living in a time, actually that's a concept that we developed much earlier than finding those people, but in a sense it is a symptom of what one can call neo-lifism, this fetishization of new technological approaches to life, this idea that we are so removed, or we are decontextualizing life to such an extent that we worship the technological approach to life rather than life itself. And this is a piece that we did in 2010, but it's based on this idea that we are living in a time where we're shifting away from our traditional idea of life, where we used to privilege form, and here you can see a natural history museum in France, where the life forms are being displayed based on the idealized form beside the human. Obviously the human is still in the realm of the cabinet of curiosity, and this is quite freaky that they still have museums in Europe where they have a Maori head, a kind of a preserved Maori warrior head, the skin of a French soldier that died in a war in the 80s, and a mummified Egyptian mummy. But the fact is that the whole idea of our understanding of life was based on the idea of those idealized forms of those animals that allow us to kind of engage with them in some way. But this is how Natural History Museum looks like, or the collection looks like. This is from the Queensland Museum in Brisbane in Australia, that's how they're collecting life. And what they're really doing here is privileging the information, the potential of the information embedded within those cells, those tissues, the DNA, to tell us something about life. But obviously, it's much less evocative, and it's totally decontextualized. Although I would argue that also the stuffed animals are, in a sense, decontextualized, but here it's much more apparent. And this fetishization of new approaches to life that only comes to form when we're dealing with kind of those iconic life forms that have been modified by technology. This is the death mask of Dolly, the first cloned sheep. And if you really want to worship and fetishize it, if you go to the Science Museum in London, they have the droppings of Dolly on display on a pedestal. You know, that's how freaked out things are. And the same with like a small sculpture of the mouse with Eron's back as if it's standing for the future of the manipulated life. But it also reaches such a degree that people who are engaged in conservation are victims of this neo-lifism. So this is a space which is called the Frozen Ark. It's a consortium of international museums and zoos that come together in order to create a frozen library of different life forms that are on the verge of extinction. An insurance policy, as they talk about it, in a sense of, yeah, you know, if we are going to destroy the Earth, at least we'll have this potential embedded within those frozen specimens, so we can one day resurrect. And actually, if you go to the website now, they've got this amazing Christmas deal for you. Yeah? For a contribution of £450 or $650 would enable you, or enable them, to store the DNA of one endangered species for the long term. Yeah? A single donation of $10,000 would allow the Frozen Ark project to survive forever. What the fuck? Forever. That's a very strange. But this idea of allow future resurrection of species. The idea that by storing those things, that would allow the future resurrection of species. The idea that we don't need to care about the environment of the panda because we've got a frozen specimen somewhere. Which makes us think that maybe we should actually connect those people with the people from Frozen Meadow that wants to grow the meat and the leather and say, hey, you know, forget about this small donation, go to the PayPal dude and get some more money and grow and have a restaurant of endangered species or grow leather out of them. Or do it to such extent that we will understand how excessive and how unrealistic this idea of decontextualizing life is. And we hope, you know, I don't think we have the $10,000, but we are going to donate that amount of money because we also want to have this customized PDF document that will have to, you know, commemorate the fact that we are just giving something. So this notion of decontextualized life was reappeared, kind of reappeared in some of our works, including this piece over here. And you can see this is a traditional kind of collection of specimens from Natural History Museum, which is peppered with those cryogenic vials, where we have specimens, obviously not frozen, but kind of this idea standing for those decontextualized specimens. But what we have there as well is this technological body. And this is a bioreactor where we actually grow living cells, so this is the only living component within this whole arrangement, but the life is so obscured by technology that most people wouldn't recognize that as being alive in any way. And the life forms that we have there are cells which are called hybridomas, and those are actually a combination of two or more different species growing together into a new type of cells which can never survive. Not just nature can never survive as a body, they can only survive in the level of cells and tissues. So you can see this is arrangement when we had this piece at the National Art Museum of China in Beijing, and it was a very interesting way in which people kind of would approach it and wouldn't really get what it is. You know, this idea of the totally abstracted life, which is embedded within this technological body. Well, obviously they can relate quite well to those idealized notion of form of the animal, but we also had those this two-headed bird which was standing for the original cabinet of curiosities, this idea that actually all of our understanding of systematic collection of life forms to some extent came from a kind of compulsive collection notion of collecting things which seems odd and strange. Yeah, that's really led to the original Natural History Museum that started as collections of oddities. How are we doing with that? Good. Okay, I think I want to tell you one last story. This is actually a work that I'm developing at the moment together with Junat, and it is something that I'm still trying to come to terms with. What we realized as we were working throughout the years, we were developing different projects that were dealing obviously with this idea of the semi-living, the idea of having those fragments of life grown within those technological environments. So we're very interested in, we are focusing with our work on the cells and tissues, the living material that we're working with, and then we shifted and we started to focus on the idea of the technological body in which those cells and tissues are growing. But what we didn't really think about as much was the context, and you can see that it's kind of coming back, and this idea of the substrate on which those cells are growing, the milieu in which the life operates, as something that we really need to pay attention to. And we're trying to figure out different strategies of dealing with it. And then last year I was invited to host a group of artists in this amazing place at the very far north of Finland, in a research station called Kilpesari, up in way above the Arctic Circle. And I decided to use that as a way to try and find the biological milieu, this idea of the substrate. My idea of going to the place was that I would find myself in a place which is so barren and arid. You know, my vision of where the place is, or that my surrounding was, that, you know, it would be ice fields and rocks and nothing else. And I said, okay, I'll try and search the origin or something there that I can use as the starting point for the growing life as a substrate. That would be the kind of the proto-substrate, if you like. And we go there, and suddenly this place is full of life. It was teeming with life. It actually had enough life there for me to survive till winter. There were mushrooms and there were berries, and I could actually go and forage stuff. You know, I didn't really need. It was quite strange, especially coming from Australia, the idea of the desert, the idea of somewhere that you'll be left for a couple of days and you just die, and realizing that actually this extreme place that I thought I'm going to was much more hospitable, especially that season, to my existence. And obviously to the existence of so many other life forms. The earth was so thick of moss and other kind of plant material that it was really hard for me to try and find what I was looking for initially, and that was this kind of proto-substrate. And then as we were walking in one of the field trips that we've done, we came across this site over here. And, you know, it looks like a fire occurred there maybe a year ago. And we were coming closer, and you see this type of stuff around, and all of this debris, and bits of tumbled metal, and other bits and pieces. And realizing that this was a crash site of a German plane, a Junker 88, from 1942. So first of all, it told me that actually my illusion of that place being hospitable for life was a bit strange, because actually this place is under ice for most of the year, and that's why this still survived and such. You know, I know that in Australia, if there's a fire the year after, and lots of stuff is growing up, and actually after 2 or 3 years you won't even see any evidence of the fire occurring. And here this is something from 1942 that still looks very much like it happened just now. But then I was thinking a bit further, because it reminded me some stories that I was thinking about. So here are the Junkers 88. This is one of the planes that crashed. You can see, you know, the noses there. And in the history of biomaterials, you learn about something which was really interesting that happened with those planes, because those planes were using a new type of plastic for their canopies. And when pilots would come back from war after crashing and have shrapnels of this plastic in their eyes and in other places, the realization was that actually the body doesn't reject those materials. And it was the first time that they were starting to realize that there are synthetic materials, man-made materials, that can actually be compatible with the body. And that was the birth of contemporary or modern biomaterials, the idea that actually we can manufacture those prosthetics. And actually, you can see here, that's the piece of plastic that I was looking at. And actually, things started to kind of mesh in my mind, because also another thing that I remembered was that when Charles Lindbergh was invited by the Luftwaffe, the German Air Force, in 1936 to 1938 to visit them, he was the first American to see one of those Junker planes. But he was also, and as you would see here in the quote, this is a quote from a paper that he wrote in 1939 at Reader's Digest. Can you imagine something that has been published in Reader's Digest? Where it talks about aviation as a way to stop. You see here the sea of yellow, black and brown. He basically said we should join the Germans, we white people should join the Germans because this is what we need to fight for or against. And we need to use our technology, our aviation, as a barrier against this invasive contamination. Yeah? So this is kind of the logic. And I promise that I'll mention Alexis Carrel's book because in 1938 when Lindbergh and Carrel were working together, Carrel writes this book called Many Unknown where he proposed the use of gas chambers to eliminate undesirable elements in human society. Yeah. So I just want to put those things together for you. So here you see them again in publication. But as I said, also this idea of the PMM, this new material that was developed at the time, was actually the basic material that we were using, or we were using a derivative of that when we approached this first scientific lab where we worked with. And I was starting to think to myself that I had to travel all the way to the far north of Finland to try and find a biological milieu, only to be fascinated by a piece of plastic, which was artificial, and was the birth of this contemporary idea of regenerative medicine, in a sense. And that really freaked me out, because when you think about it, in the very same essay, actually, the next sentence after this sea of yellow, black, and brown, Charles Lindbergh has this following quote, where he talks about the fact that aviation, using it symbolically as well as in its own rights, bring two great dangers. One peculiar to our modern civilization, the other older than history. Since aviation is dependent on the intricate organization of life and industry, it carries with it the environmental danger of a people too far separated from the soil and from the sea. The danger that the physical decline, which is so often goes with the high intellectual development, of that spiritual decline, which seems inevitably to accompany the industrial life, or that of a racial decline, which follows physical and spiritual mediocrity. And here I am, standing in front of you. Thank you. Questions? Yes.
Do you face any sort of like religious or political backlash for your work? I mean, you're obviously dealing with some fundamental like hot points in culture today, and so.
On quite a few different levels, you know, I never been threatened to such a thing. I was threatened by vegetarians, which was very strange. It was kind of funny. Again, talking to MIT, and the veggie MIT people were kind of up in arms to try and block us. When we had a show in France, I can't remember his name at the moment, but there's this guy who burns down McDonald's in France, and what's his name? Okay, I'll come back to me. He and his mob were coming to burn down the gallery, and after a long discussion, they actually came to the show and seen it, and were kind of endorsing it. To do with religious stuff, actually, I have a couple of more slides that I might be able to show you. Okay, this is a religious work that we're doing at the moment around the golem story, which we might talk about later on. This is me with a golden shovel collecting mud from under the bridge in Warsaw. We just had a big retrospective of our work in Warsaw earlier this year, and that was the time where I didn't talk about God and abortion as much as when I was in Poland. So that was coming up again and again. But the most interesting issue in regard to kind of blasphemy and religious stuff actually came across when we were doing this work with Stella, where we've grown a version of his left ear, a miniaturized version of his left ear. And this work was selected for this big art award in Australia by an external jury, and the show was supposed to be at the National Gallery of Victoria in Melbourne. And the National Gallery of Victoria in Melbourne a few years earlier was showing Peace Prize, the Oron work. And religious right in Australia got quite violent, and they were threatening people, and actually one of them got into the gallery and smashed the piece. So they were very, very sensitive, and they tried to stop us from showing this work there. So it was kind of interesting, because galleries never censor work, yeah? So they always try to find ways to stop you. They kept on asking us for totally unrealistic stuff. So initially, they basically said that, how do you work? Yeah. First, they wanted an approval. So all of the work that we're doing, we're based in a university. All of the work that we do need to be cleared by the law, by the university, by the institutional health and safety committee, but also the ethics committee, which is also a really interesting part of our practice to convince people from ethics committees that what we're doing is actually appropriate. So in this case, we got the clearance. Then the gallery or the curator asked us to have a statement that says that our work doesn't raise any ethical issues. And we said, like, what the fuck, that's really strange. and we refused to do it. And they were kind of about to pull out the work. And we went back and forth, and we decided actually with Stella to have the Salon de Réfugiés just across the road from them and have showing it there. And they realized that that might be even worse for them. So they invited us back, but they said, you can't use human cells to grow your replica of Stella's ear. And for them, it seems that it was less blasphemous to use mouse cells to grow into a shape of the human ear than having human cells growing into that shape. And we said, okay, because actually it worked really well with some of our kind of writings and our thinking of the time, where we're talking that there's no difference between, in the level we're working with, there's no difference between the human and the animal. Yeah. But, you know, the idea that this is the image of God, that somehow, you know, that Stella is the image of God, that if we are going to engage with it in some way, we're going to create some kind of a blasphemous step. We found quite appealing, you know, as well. It was quite an interesting idea about how one sees God. But in the end, and that seems to be time and time again, it seems that the galleries, the museums are way more concerned about how the public would react than how the public actually reacts. And maybe because religious people don't really get it, maybe because beside the earpiece we never really were interested in using kind of the human body or anything that represents the human body, because we believe that actually when you work with stuff that deals with the human body, there's already a very kind of established discourse around it, which is not necessarily relevant to what we're doing. And we're trying to find ways to actually force people to realize that actually there's no language to engage with what we're doing, so we're much less interested in that. But in Poland it was interesting, because in Poland, as we were showing our work, and Poland, as you know, just joined the European Union recently. And it's a highly religious Catholic state, and the relationship between state and church is actually quite strong. And they refused to sign the European Declaration for Ethics, because they believe it's not strong enough, so it doesn't fit the ideology of the church. But they couldn't come up with any alternative. So at the moment, Poland is one of the freest places in the world where one can do research without any ethics consideration, because there's no guideline or framework to actually work with it. So that's another really interesting aspect where kind of things can go a bit messy. And at the very same time, when we were showing our work, there was a big debate if the state should support in vitro fertilization. And the church was opposing it because of the issues surrounding the potential of creating excess embryos, and then what happens to them. And in a sense, that kind of links quite nicely with the whole idea of neo-lifism. When you think about those people from the conservation people that say that you can actually freeze potential of animals and bring them and resurrect them in one day, you might play into the hands of the pro-life people. Yeah. So there's some really interesting tensions that, again, I don't think, because we don't really have the language, because we don't have any kind of form of cultural articulation around it, we might find ourselves in a very strange situation. Yes?
I've got two questions for you. The one question is about Dolly the sheep. Mm-hmm. Was the problem with Dolly, I think this might have just been, I don't know how true it is, but that there's a certain aging that the cells automatic. Yeah. Was that true?
Let me qualify myself. I'm not a scientist. I never claimed to be a scientist. I have no scientific training. But I know a little bit about science and biology, because I spent my last 16 years in labs. So what I'm going to answer is a qualified answer, in the sense that I'm not a scientist and don't believe me because I'm an artist. First of all, right? But yeah, you're right. There's a thing which is called the telomere, which is a bit in the end of the chromosome, that each time the cell divides, as chip of it falls off. So this is why you have a finite amount. Most cells in our bodies have a finite amount of divisions. It's called the Havlik effect. There are two types of cells that don't have it, or they can regenerate this end of the chromosome. Those are stem cells, and that's one of the reasons we're looking at. And the other one are cancer cells, which are being referred to within the context of what we're doing as immortalized cells, because they can divide forever. So you know about the HeLa cells, those cells that were taken from an African-American woman in 1951. Those cells still growing. We went through thousands and thousands of divisions because they're actually cancer cells that don't have this self-destruct mechanism. So some people would claim that, yeah, there might be the case that Dolly died prematurely was because of the fact that the cells that were used were actually cells that already went through some kind of or some numbers of divisions, which meant that everything in the body of Dolly was, in a sense, older than her chronological age. But,
the other question I had is, to grow an ear,
yeah.
Where are the cells coming from?
Right, so we've grown Stellax ear this year, the quarter scale version, I think about 4 or 5 times. That image over here, the very original ear that we've grown in the gallery called Kapelica in UWM in Slovenia, we're actually working with a company that was working with patients that were regenerating cartridge cells or cartridge for knee reconstructions. So they were actually human cartridge cells. So in a sense, this is as close as you can get to a real ear as one can get. For the show at the National Gallery of Victoria, we had to use mouse cells, so we actually used immortalized cells, so they were actually cancerous cells from a mouse that were grown into a shape of an ear. And just to show you, I think we have... Yeah, the way we've done it was to take a plaster cast of Stellax ear, and then we used a 3-dimensional scanner to scan it into a computer, and then we miniaturized it and outputted it on a 3D printer, and created this negative shape out of silicon, and then made it out of this very special polymer material, which is a very porous material, which was then seeded with the cells. So initially the cartridge cells, then the mouse cells. We actually, in one version, we used when we had a one-act performance of killing of the ear, so we actually killed the ear as a performance, we used the HeLa cells. So each time we used different types of cells. Because the idea was to actually grow living tissue into the shape of the ear, regardless of really what the tissue was, especially after we've done, in a sense, a proof of concept the first time with the human cartridge cells.
But you were using a mold.
Yeah, exactly. There's no way you can create a three-dimensional tissue in a shape without having some kind of instructions for the cells to grow onto. So you have different types of, it refers to the scaffolds. And the only place where you can actually grow a three-dimensional collection of cells into a predetermined shape is in a uterus, as an embryo. As part of the normal development of an organism. If you are trying to do it externally from the body, you always need to somehow grow it over a three-dimensional scaffold of cells. So all of the work that we've done, all of the three-dimensional work we've done, actually involves the construction of the artificial scaffold and then seeding it with appropriate cells and using those degradable materials, so as the cells grow, the actual material breaks down, and we replace, it's like lost wax casting kind of thing. Yeah, so the living tissue replaces bit by bit the artificial support mechanism. Yes?
Do you see the oppositional push that you're getting from all these forces, I guess, taking you away from human cells as a kind of a challenge?
I'm not interested in the human. My interest is life in general. I think that being obsessed about the human is actually extremely problematic when you're starting to work at those levels. So, there's two ways one can talk about it. One can talk about it that what we choose to do to bodies, regardless if they're human or others. If we put chickens in incubators, we put babies in incubators. If we put cows in industrial farmings, we put people in concentration camps. I don't see any. From my perspective, there's no real difference. So we need to be very aware that we can't separate life just on the basis of that being human or not. Yes?
Back to the ear, I was just wondering, what's the purpose of shrinking it? Was that just to get away from...
It was both a practical thing as well as this idea, because when we worked with Stellap, so Stellap was really interested in having his third ear, and actually he's got now one. Oh, that's a killing. That was in one of the shows. But this is Stellap now. That's got nothing to do with us. So Stellap was really interested in creating an arm, a third ear. Initially, as you've seen, he wanted it on his face. Then now he's got the ear on an arm. That's nothing to do with us, but when he approached us, we decided to we were really interested in this idea of semi-living and the objects of partial life. He was really interested in at least having some kind of a gesture. This is in 2003, so, he just was able to attach this year in 2006, so, he was talking about it since he'd seen the mouse with neurons back in the mid-90s, so he was talking about having a third ear for a really long time. So it was incremental steps of gestures towards doing that. And when we decided to work together, the idea was how, it was also an experiment in collaboration, how one can have, or more than one, how two artists with two very, very different approaches and narratives can meet in one object. So it was one object with two very, very separate narratives that were following it. And we were really interested in we wanted to experiment what would happen when we use a recognizably human part. It was an homage for us also to the mouse with earrings web. And we were not surprised. The fact that that was one of the works that was treated as if it's blasphemous proved to us that we're wasting our time dealing with human form. It's not really our interest. But the idea that it was a quarter scale was because it was a gesture. It wasn't really a way of drawing Stellar's real ear that he wanted to attach. So we were that was a compromise or not a compromise, but it was a conceptual decision that was made by Stellar and us to be able to distinguish that from what Stellar aim was, which was this piece. You asked before, I was just trying to get someone else. And then, sure. Yes, over there.
For the leather project,
yes?
How exactly did the leather form? Because with the ear you have the mold, but obviously the leather is hanging, so how exactly did it form in the shape that it did?
So again, we're using let me see if I can find it. Yeah, so you can see this is, again, it's a system that we developed which is very much linked to the original organ perfusion pump by Carrel and Lindbergh as well. So again, it was a reference, a direct reference to that. And what we've done was to use another type of polymer that comes as it's like felt. So it's a fabric-like material, very similar to what we used for. That's how it comes, without the cells on it. So it's like it comes as big sheets, and then you can cut it into shape. So with the jacket, we basically cut it into a shape of a jacket. We use this drip feed system to feed the jacket, and actually when you grow skin, you want to have what's called a media air interface. So this actually proved to be quite a successful way of doing it because the nutrient solution and this material is very much like sponge, so you basically drip the nutrients on it and they disperse throughout the sponge material, this felt, and the cells are being fed as it's being done. Actually, someone asked me to comment about the arrangement here, and just to put it in context, this is a body. This is a technological body. The pump in the back is like the heart. Here you have the reservoir of nutrients, which is like the blood. And this is the gas exchange chamber, which is like the lungs. And here you have the organ, and in this case, it's a little jacket made out of a combination of mouse and human cells. So in a sense, it's kind of a minimal technological body that one can have in order to grow those cultures. But also, back to your question, we've done some experiments, and we don't really usually do it. We're not plain scientists. We're really interested more in the conceptual issues of dealing with the material, the living systems that we operate, the context, the substrate. But in this case, we were really interested in figuring out if we can grow leather, and those are some of the experiments that we've done when we grown the leather flat, and then used different tanning techniques to preserve it, and those are still preserved. So this was the most successful one. This one was just growing skin tissue with no support, and this one was using a polymer that broke down too fast and became, it didn't really work, and this ended up being very much like leather. Yeah. You want to ask a question?
Oh, actually, I was going to ask if you had. Well, I was gonna ask if you had any experience in specialized cells, like obviously nervous muscle or like mucous membrane, but you already covered that, so never mind.
Yeah, although let me put it in a different way. There's a place called the American Tissue Culture Collection. So it's a tissue bank that you can mail order cells from. And we had this Frankensteinian project where we were trying to figure out if we can actually order enough cells and different types of cells that actually constitute a body. Yeah? So if you can, all of those specialized cells, but grow them separately. But this idea that you can actually have, that there is an online collection that one can order stuff. Obviously you need a license to to do it, unless you live in Norway and then you order it to your kitchen. But you know, it's quite an amazing place, just as a way to explore what the possibilities are. And I just did another thing, because we did a project with Oron, where she was very interested in growing skin of people who had races. And that's a very French kind of approach, and to do with combining different types of cells. It's a long story. But anyway, the only way I could come to terms with actually allowing her to do such a project in my lab was to go to the ATCC, because there's still cells that have a racial designation on them, which is very strange, because scientifically it doesn't make sense. But you can go there, and in one of the fields, the search fields, you can say black skin, or Asian skin, or Caucasian skin, and get a list of cells that have this racial designation. And that's a really, really strange thing, because there's no way any useful scientific information can come out of that. But it's still appearing within this collection. So going back to our good friend Lindbergh, the sea of brown, yellow, and black, and we should look at purity. Yeah. Yes?
I consider myself a non-religious person.
Okay.
So I start with that. I'm not going to be one of those that like freedom. But one way or another, I don't know if it's because of like education or whatever. Sometimes when I see your work, it's like it makes like a lot of impact on myself. And I'm wondering if you can tell us a little bit of how do you feel in terms of like the spiritual thing, not like how do you feel in terms of like maybe trying to be God. But I'm not gonna. I mean, it's just like
yes.
You know what I mean?
No, that's a great question. I think there's a couple of things. I think the reason why you feel uncomfortable about it exactly points to the fact that we don't have the cultural articulation, the cultural way of engaging with it. So the only response that we have is a gut feeling. It's a visceral response. That I think it's extremely appropriate. Now,
and I'm not religious. No.
Yes, I hear. But I engage in things that makes me feel uncomfortable. As an artist, I think it's really important for me to constantly kind of look at what makes me uncomfortable and then try and kind of, in a sense, report back. Because, in a sense, I'm a messenger. I spend time in labs, but whatever I do is extremely trivial within the scientific context. Maybe I reappropriate, I recontextualize it, but the stuff, the actual procedure, the tissue culture, for example, and that's another really interesting thing, tissue culture is exactly 100 years old. The first experiment by Alexis Carrel was in 1912. It's not a new technology, but it still makes us feel strange about it because we never really articulated it, we never really talked about it. Another thing I want to say about, I don't like to talk about myself too much, although this was like one of the most revealing talks I ever had about kind of me identifying with the decline of Western civilization, according to Lindbergh. But I'm a very carbon life-based biased. I think there's something special about life, which cannot, we don't even need to resort to metaphysical arguments to say that there's something special about life. Maybe just because we are carbon life forms ourselves. And as I said, it's whatever we choose to do to life, we choose to do to ourselves. So I would like to believe that there's something special about life which cannot be reduced to just a merely materialistic perspective, but also can be resorted without any metaphysical argument. And this is kind of my lifelong search. So this idea of what can one argue secular vitalism is really what I'm looking at. And I don't have an answer. I'm not a philosopher, I'm an artist. I just do my shit. It's not... and I'm trying, that's why I share those stories. I want other people to engage with it as well. Yes?
Your work for me seems to problematize factory farming and also the devaluing of life in general. So I found it particularly interesting that vegetarians were up in arms at MIT. I'm just wondering if there's anything that you're advocating in particular, like as far as the diet or fashion or otherwise, or if you're just trying to provoke a dialogue, and because on the one hand you're engaged in attempting to create these foodstuffs and articles of clothing that may or may not be victimless.
Yeah. Okay, thanks for that. I think what I'm trying to advocate is what I refer to as informed hypocrisy. So you have to be aware, I can't engage, self-righteousness is something I can't deal with. I think by being, I'm totally implicated. And when people come to my lab, when people come to my workshops, the first thing I say is like, you are going to be implicated. There's no way you can remove yourself, there's no way you can unexperience or undo what you're about to do, which is things like genetic engineering and like working with cells, with human cells and whatever. So there's no way I can say to the scientists I work with that what you do is wrong. I can say, you might be bullshitting us and telling us you're going to save the world while actually hiding the victims, or you're going to, like this modern meadow people, that you're trying to get money out of people in the false pretense that you are actually solving any problems while actually creating other problems. I want to expose those types of things. I want to engage in kind of informing the hypocritical aspects of human existence. I think that, there's no form of ethical living that can be fully self-righteous. And I think that our existence is constantly in the expense of other beings. We need to be aware of it. We can't stop existing. We need to be aware of it, and we shouldn't also be too self-righteous to such an extent of telling other people that what you're doing is wrong. So, but you can always shine a mirror at them, and that's really what we do.